Good morning, students. Myself, Dr. Manoj Kumar Pandey, Assistant Professor, Department of Zoology, Ganga Singh College, Chhapra. The topic of current discussion is the secretion of thyroid gland. That is an important segment of fourth paper of the second year Zoology honors. So, first of all, the thyroid gland, which is located immediately below the larynx on either side of and anterior to the trachea, secretes two significant hormones, thyroxine and triiodothyronin, commonly called T4 and T3, that have the profound effect of increasing the metabolic rate of the body. It also secretes calcitonin. About 93% of the metabolically active hormones secreted by the thyroid gland is thyroxine and 7% is triiodothyronin. However, almost all the thyroxine is eventually converted to triiodothyronin in the tissue, so that both are important functionally. The function of these two hormones are qualitatively the same, but they differ in rapidity and intensity of action. Triiodothyronin is about 4 times as potent as thyroxine, but it is present in the blood in much smaller quantities and persists for a much shorter time than does thyroxine. The thyroid gland is composed of large number of closed follicles, that is 100 to 300 in numbers, filled with a secretory substance called colloid and lined with colloidal epithelioid cells that secretes into the interior of the follicle. The major constituent of the colloid is the large glycoproteins thyroglobulin, which contains the thyroid hormones within its molecule. Once the secretion has entered the follicle, it must be absorbed back through the follicular epithelium into the blood before it can function in the body. Iodine, the form of normal quantities of thyroxine, about 50 milligrams of ingested iodine in the form of iodides are required each year. Now, what is iodine pump? It is also known as iodide trapping. The first stage in the formation of thyroid hormone is transport of iodides from the blood into the thyroid glandular cells and follicles. The basal membrane of the thyroid cells has the specific ability to pump the iodide actively to the interior of the cell. This is called iodide trapping. Now, the oxidation of the iodide ion. The first essential step in the formation of thyroid hormone is conversion of the iodide ions to an oxidized form of iodine, either nascent iodine. This oxidation of iodine is promoted by the enzyme peroxidase and its accompanying hydrogen peroxide, which provide a potent system capable of oxidizing iodides. The peroxidase is located either in the apical membrane of the cell or attached to it, thus providing the oxidized iodine at exactly the point in the cell where the thyroglobulin molecules issue forth from the Golgi apparatus and then through the membrane into the stored colloid. Now, iodination of the tyrosine and formation of the thyroid hormone, organification of the thyroglobulin. The binding of the iodine with a thyroglobulin molecule is called organification of the thyroglobulin. It is oxidized iodine, even in the molecular form, will bind directly but slowly with the amino acid tyrosine. But in the thyroid cell, the oxidized iodine is associated with an iodinase enzyme. Therefore, almost as rapidly as the thyroglobulin molecule is released from Golgi apparatus, as it is secreted through the apical cell membrane into the follicle, iodine binds with about 1/6 of the tyrosine amino acids within the thyroglobulin molecule. Now, storage of thyroglobulin. After the synthesis of thyroid hormone has run its course, each A thyroglobulin molecule contains 1 to 3 thyroxine molecules and average of 1 triiodothyroxine molecule for every 14 molecule of thyroxine. In this form, the thyroid hormone are stored in the follicle in an amount sufficient to supply the body with its normal requirement of thyroid hormone for 2 to 3 months. Therefore, when synthesis of thyroid hormone ceases, the effect of deficiency are not observed for several months. The major hormonal product of coupling reaction is the molecular thyroxine that remains part of the thyroglobulin molecule. One molecule of monoiodotyrosine coupled with one molecule of diiodotyrosine to form triiodothyronin, which represents about 1/15 of the stored hormone. Now, the transport of thyroxine and triiodothyroxine to the tissue. On entering the blood, all but a fraction of 1% of the thyroxine and triiodothyronin combine immediately with several of the plasma proteins. They combine mainly with thyroxine binding globulin, but much less so with thyroxine binding prealbumin and albumin. And the thyroglobulin itself is not released into the circulating blood in measurable amounts. Instead, thyroxine and thyroglobulin are first cleaved from the thyroglobulin molecule, and then these free hormones are released. So, here are some important functions. Effect of thyroid hormones on mitochondria. When thyroxine or tyronin is given to an animal, the mitochondria in most of the body increase in size as well as number. Furthermore, the total membrane surface area of the mitochondria increases almost directly in proportion to the increased metabolic rate of the whole animal. Therefore, it seems almost to be an obvious deduction that one of the principal functions of thyroxine might be simply to increase the number and activity of mitochondria, and they in turn increase the rate of formation of adenosine triphosphate, that is ATP, to generate cellular function. However, the increase in the number and activity of mitochondria could be the result of increased activity of the cells as well as the cause of the increase. Now, effects on carbohydrate metabolism. Thyroid hormone stimulate almost all aspects of carbohydrate metabolism, including rapid uptake of glucose by the cells, enhanced glycolysis, enhanced gluconeogenesis, increased rate of absorption from the gastrointestinal tract, and even increased insulin secretion with its resultant secondary effects on carbohydrate metabolism. Effect on fat metabolism. Essentially, all aspects of fat metabolism are also enhanced under the influence of thyroid hormones. Because fats are the major source of long-term energy supplies, that fat stores of the body are depleted to a greater extent than are most of the other tissue elements. Effects on plasma and liver fats. Increase thyroid hormone decreases the quantity of cholesterol, phospholipids, and triglycerides in the plasma, even though it increases the free fatty acids. Conversely, decreased thyroid secretion greatly increases the plasma concentration of cholesterol, phospholipids, and glycerides, and must always cause excessive and deposition of fat in the liver as well. So, the third effect, that is effect on vitamin metabolism. Because thyroid hormone increases the quantities of many of the enzymes, and because vitamins are essential parts of some of the enzymes or coenzymes, thyroid hormone causes increased need of vitamin. Therefore, a relative vitamin deficiency can occur when excess thyroid hormone is secreted, unless at the same time increased quantities of vitamins are available. Effects on basal metabolism, that is BMR. Because thyroid hormone increases metabolism in almost all cells of the body, excessive quantities of the hormone can occasionally increase the BMR rate to 60 to 100% above normal. Now, effect on body weight. Greatly increased thyroid hormone almost always decreases the body weight, and greatly decreased hormone almost always increases the body weight. These effects do not always occur because thyroid hormone increases the appetite, and this may overbalance the change in the metabolic rate. So these are the functions of thyroid hormone, and I think this is the final presentation. Thanks.